Thank you. Good morning, everyone. Before I kick off, I'd like to say thank you to ARM for the opportunity to come here today to tell you about Carisma and our engineered macrophage platform. Before I start, as a newly minted public company, I'd like to draw your attention to our forward-looking statements. So Carisma Therapeutics is pioneering the development of a new class of cell therapy, engineered macrophages. Initial focus is in oncology, but we see this as a broad platform that could address a number of devastating diseases. The company is about 5 years old. We're based in the Silicon Valley in Philadelphia, founded by pioneers in cell therapy out of the University of Pennsylvania. And since the inception of the company, we've really had two key goals. The first is to continue the development of the proprietary platform that was started at Penn. So we're now working in autologous ex vivo cell therapy. That's our lead program in the clinic. But we also have earlier programs in allogeneic. And then in addition to that, last year, through our partnership with Carisma Therapeutics, we started to develop in vivo targeted macrophage therapies as well. Underpinning all of this is a significant IP portfolio. We now have 15 granted patents, 40 pending, and we have coverage essentially for any CAR macrophage, any CAR monocyte, irrespective of target, starting cell, how that cell was engineered. So I think that makes us quite unique in the cell therapy space. The other key objective for the company was that we recognized very early on as a first-in-class cell therapy. No one had ever taken an engineered macrophage into the clinic before. It was going to be vital to create clinical data. Obviously, the translational models in oncology aren't always predictive. So last year, we started the first ever study of an engineered macrophage with our lead HER2 program. We provided an update at SITC, which I'll share with you today. But essentially, we were able to demonstrate feasibility, a good safety profile, and the mechanism of action for this first ever study. In addition to that, it was a big year for us last year. We entered into the collaboration with Moderna at the start of the year. The opportunity really here is to work with one of the world leaders in mRNA and LNP to combine their expertise, their platform, with our know-how, our knowledge, our proprietary technology and engineering in vivo targeted macrophages. So taking an off-the-shelf approach, but developing an autologous cell therapy within the patient's own body. As part of that partnership, we received $80 million up front, full funding for up to 12 programs, all in the oncology space, as well as milestones and royalties. In addition to that, we also, at the start of March, entered into a reverse merger with Sesen Bio. This provided a public listing for the company on the NASDAQ Global Market, along with approximately $105 million from the merger and a concurrent financing. So at this point, the company is well funded through into 2024, and critically through the remainder of our HER2 program and a number of clinical and preclinical value inflection points. So why macrophages? I found it very, very interesting coming to the conference over the last few days that when I joined 4 years ago, I was often the only person presenting at a conference on macrophage therapies. It's great to see the presentation from Resolution, from Blue Rock yesterday. And we believe that this is really the start of macrophages becoming essentially the third leg of the stool, along with T cells and NK cells. The founders of the company really chose to focus on macrophages as it's truly the ultimate multitasker of the cell therapy world. Macrophages play a variety of different roles depending on where they're located around the body. So obviously we're focused on oncology initially, engineering macrophages to try and flip the tumor microenvironment to make it pro-inflammatory. But they also play a role in immune response, in tissue regeneration, in liver fibrosis, a number of other diseases. So we believe it really gives us a broad platform to address a number of different devastating disorders. The reason the founders of the company decided to focus on oncology and specifically solid tumors initially was essentially because of the unique mechanism of action of macrophages within the TME. Really three key challenges which have hampered the development of cell therapy in solid tumors. The first of those is trafficking. T cells and NK cells don't naturally traffic into the tumor microenvironment. Secondly, the immunosuppressive TME shuts those cells down. And then thirdly, unlike a number of blood malignancies, solid tumors are incredibly heterogeneous. So you can't just target one antigen within the tumor. You need to be able to mount a broad response against multiple neoepitopes. Macrophages get around this essentially through their innate mechanism of action. They're one of the only immune cells that is actively drawn into the tumor. In the case of most of the cells within the tumor, the macrophages, they're present as tumor-associated macrophages. Obviously, what we want to do is the opposite. We use that natural trafficking ability essentially as a Trojan horse mechanism. We engineer our cells to be pro-inflammatory, so the opposite of the TAMs. We add a CAR into the mix, so they traffic into the tumor. They form a synapse with the tumor cells. They phagocytose, clear the tumor. At the same time, they are essentially play a key role in modulation of the TME. So by flipping them into a pro-inflammatory state, they'll release cytokines, chemokines, warming up the cold tumor microenvironment. And then lastly, as one of the only professional antigen-presenting cells, they're able to chew through the tumor and present those multiple neoepitopes out to the adaptive immune system for further destruction. So we believe it's really this unique triple mechanism of action which potentially holds the key to unlocking the challenge with solid tumors. As I mentioned, we've invested a lot of time and effort over the last 5 years building our proprietary engineered platform. One of the reasons we believe there hasn't been a big explosion in macrophage companies is simply they're very difficult cells to engineer. So the breakthrough at Penn that led to the foundation of our company was the discovery of a novel viral vector, Ad5F35, and this, along with VPX lenti, which is another viral vector we retain exclusive rights to, are the only two vectors that we're aware of which are effective at engineering these cells. So in addition to both of those, which we use for ex vivo cell therapy, now through our partnership with Moderna, as I mentioned, we're also using mRNA LNP to target these cells in vivo. We can engineer them into either pro-inflammatory or anti-inflammatory phenotypes, depending on the disease state we're trying to address. And then we've been able to demonstrate the delivery of a variety of different payloads. Obviously, we're starting with CARs, but we can deliver enzymes, hormones, different peptides, pretty much whatever we want to into these cells now. This is our pipeline. As I mentioned, we recognized it was going to be critical to enter the clinic to start to build a baseline of data to understand how these cells are functioning within humans. We now have a fairly extensive program for our lead HER2 asset, starting off with the monotherapy, which we started last year. Earlier this year, we initiated a pembrolizumab combination study, and then we will start a HER2 CAR monocyte study early next year as well. In addition to that, we have a next-generation mesothelin-targeted CAR program. This will use our next-generation CAR construct. The HER2 CAR was designed over 5 years ago, so we know that we can significantly improve on that first-generation design, and that's what we're going to put into the mesothelin program. We also have an early discovery stage program targeting PSMA. In addition to that, we've been working with Bruce Blazer, who's really one of the pioneers of iPSC cell therapy at the University of Minnesota to develop iPSC-generated macrophage therapies. And then through the work with Moderna, we've now initiated work on 4 different both solid and blood malignancy targets, all in oncology. I'll provide a quick summary of the initial HER2 data that we shared at SITC. Very briefly, the manufacturing process for our macrophage product is fairly similar to a T cell. The only difference for us is that we don't lymphodeplete. Obviously, we want to retain as much of that intact immune system as we can. We treat the patients with sub-Q GCSF. We pre-mobilize them, harvest the PBMCs, isolate CD14 positive monocytes, differentiate them into macrophages, deploy our adenoviral vector to deliver the CAR. It's about a 7-day vein-to-vein time, about 3.5 weeks. 7-day processing time, 3.5 weeks vein-to-vein time. With the CAR monocyte product, we hope to take that down to around 1-day processing time. In terms of the data we shared at SITC, we were able to demonstrate feasibility. We were able to make large volumes between 2 to 4 billion cells for most of the patients we treated. Each of those cells had high CAR expression, good purity, good viability. This was the first time that anyone had done this from a patient as opposed to a healthy donor. So we were very pleased with the results that we had there. The product was well tolerated. We didn't see any dose-limiting toxicities, no severe CRS, no neurotoxicity, cardiotox. So we're very happy with this initial safety profile. In terms of best outcome to date, we've seen in about half of the patients stable disease as the best outcome. And then finally, in terms of mechanism of action, so one of the things we invested heavily in was a whole array of correlative studies to really understand whether the mechanism was playing out. And what we were able to see there was really across the board from trafficking. In 8 out of 9 patients, we were able to detect the CAR, so we know it's getting into the TME. We were able to track the changes within the TME, warming up of the tumor, release of cytokines, chemokines, infiltration of effector cells, T cells, dendritic cells, and then specifically new tumor-reactive T cell clones within the tumor microenvironment, suggestive of antigen presentation and triggering the adaptive immune system. So at this point, we are confident that we have a product which has a good tolerability profile and is doing what it's supposed to do, but we recognize that we need to go further on this. So the key next steps for the HER2 program, as I mentioned, are that we have initiated the PD-1 combination study. We do believe in these very late-stage, heavily pretreated patients that the addition of a checkpoint inhibitor will be key, essentially to unlocking the T cell response. The macrophages can prime the T cells, they can recruit them in, they can present antigen, but if the patient's T cells are exhausted or they have next to no T cells, which we see with a lot of these patients at baseline, it's not going to re-energize the T cells. So we need to put in a PD-1 to do that. The next step will be the CAR monocyte. As I mentioned, we can generate far more of these cells. We can go from a dose of around 2 to 4 billion with the macrophage to 10 to 12 billion, so significantly increase the dose. In addition, these are smaller spherical cells. We believe there's an opportunity for them to traffic better, and so that combination of better trafficking, increased dose, we also believe could lead to a higher level of efficacy. We have the next generation construct, as I mentioned, with the mesothelin program, and then novel modalities. So we're super excited about the early allo programs and the in vivo programs with Moderna. In terms of where next after oncology, I'd say to date we're pretty much 90-95% focused on oncology, but we do see a significant opportunity here to go into other areas. We have been working on engineering macrophages to address liver fibrosis. That work is in-house. We also have a partnership with the University of Pennsylvania to develop CAR microglia targeted against beta amyloid, where they can clear the amyloid deposits and do that in an anti-inflammatory fashion. So we think that's a very interesting approach. We've also been working on the development of switch receptors for immune disorders, where they can sense either an anti-inflammatory environment in terms of the TME and mount a pro-inflammatory response, or do the opposite with a anti-inflammatory environment, a pro-inflammatory environment, and then dampening down the immune response. In terms of where we're looking to partner, oncology, we have the Moderna partnership. That's the main focus for us. I think in some of these non-oncology areas, that's really where we see the opportunity. We recognize that as a small company, we can't do everything. We can't be expert in every area. So we see a big opportunity to partner with companies who have the disease area expertise. These are our milestones going out over the next 18 months, so delivering the key clinical milestones, which I'd mentioned for the HER2 program, continuing to progress the engineered macrophage platform, and then advancing the pipeline. So to summarize, Carisma is the leader in engineered macrophage therapies with a number of programs in oncology and beyond. We have a broad platform, both in autologous, allogenic, and in vivo cell therapy. We have a validating partnership with Moderna, an experienced team, and a number of value inflection milestones over the next 18 months. And with that, thank you.